So the second part of this topic is actually on cancer and transcriptional control because we know that proto-oncogenes are involved in transcriptional or transcription. So many transcription factors, especially those that are proto-oncogenes or tumor suppressors, usually, well, the function is to help regulate the cell cycle and because if it is as such, so it would determine how large a cell will get and when it can divide into two daughter cells or not. So any alterations in cells that give rise to cancer can therefore affect the transcription and control of gene expression. So mutations that activate transcription factors such as increased phosphorylation can also increase the binding of a transcription factor to its binding site in a promoter. So this is actually could be a good thing. Most of the time it's not a good thing because it could lead to increased transcriptional activation of that gene that results in modified cell growth. And another alternative is that a mutation in the DNA of a promoter or enhanced region could also increase the binding ability of a transcription factor. This same thing could lead to increased transcription factor and aberrant gene expression that is seen in cancer cells. So another misregulation of oncogenes and tumor suppressor genes is associated with the increased cellular growth and survival during early tumorigenesis. What happens is that beyond their well-established role in primary tumor formation, multiple oncogenic transcription factors are now known to also regulate metastasis. For example, these are some of the examples. c-MYC is actually a basic helix loop helix lysine zipper transcription factor that is dysregulated in 30% of all human cancers. So remember our helix and also lysine zipper. So within the genes regulated by c-MYC, it basically comprises of about 15% of the genome, and they are mediators of cell cycle control, stem cell maintenance, and apoptosis. Remember, apoptosis, programmed cell death, which is very important. So in addition, MYC has also been linked to elevated invasion and migration through increased expression of osteopontin, OPN, and ROA, and the induction of the epithelial mesenchymal transition. Thus, MYC also represents a class of oncogenes that function in both primary tumor formation and metastasis. So, well, I'm not going to eukaryotic cells. Okay, this is the same as what we have covered before. So you can go on and read about it and probably could just associate with how along the way each of the steps up until step number 5 could be affected by oncogenes. So in terms of the c-MYC just now as I was saying it has a lot of function for cell cycle, apoptosis, metabolism, dynamics, and so forth. Or basically it is the one that controls cell growth or involved in cell growth. So if there is a low c-MYC what happens is that it could pause the relief of the active genes. And the other or the opposite of when you have high c-MYC is that it could also pause the release of the active genes. How do they do that? Because transcription amplification for genes within the cells, the eliciting of gene expression program is determined by the c-MYC when it is bound to the promoter. So in low c-MYC, what happens is that, well, it will reduce or basically produce the gene as supposed to be or basically as required by cells just enough, simply said. But in high c-MYC what happens is that it would produce too much of the gene which is of course not really desirable. So what happens is that for low c-MYC usually there should be not that much of a problem. The cell or basically a part of the tissue could still survive. But for those with high c-MYC what happens is that tumor cells will grow uncontrollably and would proliferate which could lead to cancer. So researchers have been investigating how to control the transcription activation of gene expression in cancer. So this could be done by identifying how a transcription factor binds or well finding a pathway that activates where a gene could be turned off, which could lead to new drugs or new ways to treat cancer. That's what they desire it to be. So, for example, in breast cancer, so many proteins are overexpressed. We know that because usually it will create lots of lumps, well, which basically makes the breast to be removed. This could lead to increased phosphorylation, which is a key transcription factors that increases transcription. And another example of overexpression of the epidermal growth factor receptor or EGFR is a subset of breast cancer cell. And the EGFR pathway activates many protein kinases that in turn activate many transcription factors that control genes involved in cell growth. So this later on could become something which is very huge and uncontrollable. So new drugs that prevent the activation of EGFR have been developed, and of course is being used to treat these cancers, still in its infancy still. Now, how about cancer and post-transcriptional control? So stability and distribution of different transcripts is, we know, regulated post-transcriptionally by means of RNA binding protein (RBP), and RBP control various steps and rates of transcripts, as we know. Okay, so it basically is involved in alternative splicing, nuclear degradation, okay, in the exosome, the processing, export, sequestration, okay, degradation, okay, and of course translation later on to protein. So changes in post-transcriptional control of a gene would of course result in cancer, because certain specific cancers with altered expression of microRNA, okay, could occur. And if it is basically just, even though it's just basically an altered expression in the microRNA, okay, but it is, well, the cancer will be something which is very specific, and it could be hard to find the cure for it, okay, because we know that, okay, microRNA can actually bind to the 3' UTR or 5' UTR of RNA molecule, okay, to degrade them. So if there is an altered expression, okay, what happens is that probably it couldn't bind properly. Most of the time it couldn't bind properly, and it could degrade them. What happens is that they would stay in the cell, okay, which means that whereby they should be eliminated, but they stay and would cause cancer. So overexpression of this microRNA could be detrimental, of course, to normal cellular activity. So an increase in many microRNA could dramatically decrease the RNA population, leading to a decrease in protein expression. And this has been studied and has been proven. So several studies have demonstrated a change in the microRNA population in specific cancer types. And it appears that the subset of microRNAs expressed in breast cancer cells is quite different from the subset expressed in lung cancer, or even from normal breast cancer. So that means that basically microRNA cause specific types, okay, of cancer, and this cause, again, different subset of expressed microRNAs. So these types of studies also suggest that if some microRNAs are specifically expressed only in cancer cells, they could be a potential drug target. And it is also suggested that new drugs that turn off microRNA expression in cancer could be an effective method to treat cancer. But again, okay, they are still trying it out. So this is basically formation and function of microRNA. So I'm not going to dwell too much, okay, you can refer to your notes, okay, you can read on about it. Now, how about cancer and post-transcriptional control? So basically what happens is that, okay, in post-transcriptional control, okay, the one that we have been discussing, okay, early on, so we know that, okay, it could be caused by microRNA, and if it is caused by microRNA, it could cause specific types of cancer. And this is, if you were to look at the diagram A, B, and C, okay, what happens, okay, is that, okay, you get, okay, for the A is basically what it should be in a normal situation, okay? But okay, B is what happens, okay, whereby there is a mutation, okay? A mutation basically which cause an increase, okay, in the microRNA genes, and of course okay, increase in the pre-microRNA in the nucleus. So what happens is that, okay, there are lots of microRNA duplex, and this, okay, would deactivate tumor suppressor gene, which means that, okay, it basically will not, microRNA, even though it is in abundant, it basically more or less hinder tumor suppressor genes, which means that tumor suppressor genes couldn't function. So if tumor occurs, it would basically proliferate or basically divide, okay, without control. So the other situation, okay, is that whereby there is also a mutation, okay, but a decreased number. Okay? So a decreased number, what happens is that, okay, it would affect oncogenes, okay, whereby, okay, oncogenes, okay, would cause high expression of oncogenes, and we know that, again, high expression of oncogenes, okay, would eventually lead to cancer. All right, so I'll leave you to that, and we'll move on to the third video on cancer and translational or post-translational control.